All right. So I think we left off here with this idea of a redox tower. So you can see the reducing potential in black over here. Whoops, let me shift this a little bit. All right. You see all of that. So if it's above the zero, what's going on with the reduced partner? If it's above zero, does it like to give off electrons or accept electrons? Above the zero. You're above the zero. So going here, which one, H plus or H2 is reduced? Which one's reduced? Has its full complement of electrons? H2, H2. So does H2, it's in the negative portion of this reducing potential scale. So does that mean it likes to give off electrons or accept? Donate. Yep, it likes to donate off electrons. I often think of hydrogen is really happy to don't H2 is really happy to donate off electrons. I often imagine it kind of like Tinkerbell, with the magic wand and the pixie dust, where I imagine the pixie dust is a bunch of electrons that H2 is giving off. And she's so happy to do that. All right. You get below the zero and into the positive end, then it's flipped. Then what you see happening is the molecules that are to the left of the slash, the oxidized versions, they're really happy to accept. And oxygen's at the bottom because, as far as we know, oxygen is the best electron acceptor. And I often think of oxygen as the Incredible Hulk that hulks out if he doesn't get his weight. And so oxygen wants electrons. And if there's not some available, then oxygen sometimes can get a little testy about it and start to create free radical situations. That can happen in your mitochondria. Hopefully it's not happening right now, but it could be happening. And so the Hulk likes to smash. Oxygen likes to destroy other things around it by creation of free radicals if it doesn't get its fair share of oxygen. So oxygen is an excellent electron acceptor. All right. Then we looked at the idea that you have, they're all written as pairs, the redox pairs. And so everything to the left of the line, no matter where it is here, everything to the left of the slash is the oxidized state of the molecule. Everything to the right of the slash is the reduced. So if you're a reduced molecule, that means you have some electrons you can donate. If you're oxidized, that means you just lost some. You need to gain some back to become reduced. All right, so we've been looking at these ideas. Last time we did work through some examples of who would be donating, who would be accepting. So really, this is just, I like to think of it as a game the microbes are playing, where they understand the rules, they need to abide by the rules, but they just try to create a situation where they can react these molecules together that they're surrounded by, take them in, react them in such a way that, what is going on here now? That they can, there we go, get it to convert to ATP. They can create a situation where they can then synthesize ATP. So ATP doesn't stick around for very long. It gets made, it gets used, it gets made, it gets used. And so they need to be constantly doing some kind of biochemical reaction in order to keep their ATP levels going. And ATP is the main one, but there's others. So pyruvate is also pretty good. So are anything that has an anhydride bond, essentially, that when you break that anhydride bond, it provides some nice energy. That's, ATP has those triphosphates on there. So basically they're running all of these reactions, these redox reactions, so that they can get their ATP made. But there are some others. So this little table here gives some other options. It's not a complete table, but does show you some options. So the little anhydride bond, that little squiggle they always put between the O and the phosphate. When you break that bond, it does release some nice energy. That's why ATP works. It's able to release that nice energy. So you need to have a lot of ATP made through the day so you can use it for things. Did I go over yet? I can't remember. I think I did, right? -32 is the number I want you to memorize. We round up. So it's on this table, it's showing ATP is -31.8. We just round it up to -32. So in order to synthesize ATP, you have to create some kind of biochemical pathway that will yield at least -32 kilojoules per mole. And then you can get some ATP from that process. So when you think about fermentation pathways, going back to those, again, this is glycolysis is an example of fermentation, and it's a substrate level fermentation. Almost all fermentations are substrate level phosphorylation. So the way they work it is fermentation reactions. They have these chemicals available around them. They take them in, start running these pathways, and then they have the phosphate transferred from one substrate to ADP in order to get ATP. So you've been through this more than once. So remember now, glucose, the starting molecule, is C6H12O6. And then when you end up down with pyruvate or any of the stage 3 products, it's not a complete breakdown to CO2. So microbes can do this glycolysis and they'll get 2 ATP out of it. Remember, there's maybe 2 of everything in this pathway. So they make 4 ATP here, they use up 2, so they net 2 ATP. Okay, that's okay, it keeps them alive. But one reason why they don't get a huge payoff is nowhere in here is oxygen a terminal electron acceptor. That's not happening. And you don't have complete breakdown with CO2, so you're not getting as much bang for your buck in that regard either. So we say when any fermentation pathway that you look at, what we say is that it's a partial breakdown. A full breakdown would be that it went all the way to CO2. That's all you're left with is carbon dioxide. A partial breakdown means you have lactate or ethanol or something like that. It's not a complete breakdown. And you don't have a terminal electron acceptor like oxygen. You also see fermentation happening in anoxic environments where there isn't oxygen available. And so the types of microbes that can handle those conditions are either facultative anaerobes. So facultative means they can work with oxygen if it's available, and they're happy to do so. But if their environment starts to go anoxic, they're like, I'll switch to something else. They could switch to fermentation, but there's a better option that we'll talk about than that. So hold that thought. And then fully anaerobic microbes that can't stand oxygen, like they didn't evolve in the presence of oxygen, oxygen's toxic to them, then you're a full-on obligate anaerobe. And so those types of microbes can do fermentation quite regularly, plus some other things that we'll talk about. So the goal, really, when you look at all of these fermentation pathways, and remember, microbes are really, really good at doing a bunch of different fermentation pathways, depending on their environment and what you feed them. So the goal is to get your ATP through substrate level phosphorylation. This is a kind of in words, what's really happening there. And when you run that substrate level phosphorylation, when you cleave off the phosphate off of one of those substrates, it provides enough energy for that phosphate to then bond with ATP or ADP in order to make your ATP. So it's just a game they're playing. They're like, okay, what do I have around me? Oh, I got some glucose. Great, I'll run glycolysis. And in that process, I'll be able to transfer some phosphate from one of these intermediates onto ADP. So in the brewing industry, when you have your yeast making ethanol, you feed them sugars, and depending on which company, sometimes they'll spike in some interesting like plant hormones to get the grains to partially break down, so the starch hasn't, or the yeast has an easier time to attacking the starch breakdown products. But anyways, you deliberately feed them certain types of carbohydrate in order for them to really pump through fermentation to get you that ethanol. And so in the brewing industry, you know, they're fed very particular carbohydrates to maximize their ethanol output. But in natural settings, they're just working with what they have around them. And so they're constantly assessing what kinds of molecules do I have? What should I run? Should I do fermentation? Should I do a terminal electron acceptor with oxygen, something else? And so here they're looking at fermentation idea, where the substrate level phosphorylation is involved to get you your ATP. And then they don't really care about like the yeast that's making ethanol in the brewing industry. They don't care about the ethanol. Okay, that's not their goal. We capitalize on that, and it's $1 billion industry, but the yeast isn't doing anything with that ethanol. They just excrete it out as waste. So it's actually toxic to them. As I mentioned, when it builds up in those giant copper vats, you have to get that out of there for the yeast to continue to live. So if they're not going for the ethanol, like if the yeast, if they just excrete that out, you know, it's not their goal. And that's just waste to them. What they're really trying to do is create a situation for substrate level phosphorylation. And depending on the microbe, you got all kinds of options. And certainly we're not going to go through and have you memorize all of these different fermentation pathways, because that would take forever. But this is a table showing some options. And you can see, you know, the substrate starting product, starting molecules, and then the products. And you can see there's all kinds of different microbes that play this game in order to end up with a fermentation reaction. But if you look at it carefully, you can see that all of the products, they're not all CO2. There is a partial breakdown of what you started with. There might be some CO2, but it's not all CO2 that it's broken down to. So if you look up some definitions of fermentation, this is often how they will phrase it. Fermentation is the completion of catabolism without electron transport system and without a terminal electron acceptor. They should have, I like also that they should have put, and not a complete breakdown of your starting carbohydrate. So most of the time when you look at fermentation pathways, it's pretty much substrate level phosphorylation. There's one exception I'll talk to you about, but most fermentation pathways, it's the substrate level idea, where one of the intermediates in the pathway has a phosphate and it will get donated off to ADP. So it's not a huge amount of ATP. I've been trying to stress that. You don't really get that much ATP. So if you're a microbe that's doing glycolysis as one of your only ways to get ATP, that means you need to eat a lot. You need a lot of glucose in order to get enough ATP to survive and be able to reach the point where you can reproduce. And so that's why in like the brewing industry, they really have to feed their yeast a lot of carbohydrate in order to get enough of the ethanol produced. So, you know, when you look at all these fermentation pathways, there's different end products. You know, there's lactate, there's ethanol, there's butanediol, there's all kinds of other options as some of the final products. And when you really, really look at what's going on here, there's a key like rule of the game that you need to think about. So fermentation, the rule is you have to have a balance of oxidation and reduction. Just by the laws of chemistry, you've got to have that oxidation and reduction going hand in hand. Okay. But basically what that means is when you look at the entire pathway from start to finish, the electron number that you started with, so the substrates, electron numbers, how many electrons are there with the substrates, has to equal the number of electrons that you end up with. For the products, they have to equal out. And that's to serve this rule, so that you have the same amount of oxidation and reduction reactions going on. So by default, by definition, that means you have to have the same number of electrons that you ended with than what you started with. They have to equal out. And if microbes know this, they know this rule to the game. And so what happens is that they will play the game. They'll like, okay, well, I can move electrons from this substrate. I can put it onto this thing and it'll all equal out. So I'm good. I'm following the rules. And that's basically why they end up with these different fermentation products and different fermentation pathways. They're just trying to play this game and follow this key rule. And if they can do that and start off, with, in the case of glycolysis, it's typically glucose coming in. Well, then they can play this game, and maybe they'll take the fermentation to lactic acid or maybe to ethanol. But that's really what's giving rise to these different products that are then excreted, because once they have, they adhere to this rule, once they've done that, then they just excrete out the product. They're like, okay, we're good. We have everything good here with the electron number. I'll just excrete out that lactic acid or the ethanol. So some people might refer to it as redox balance. That's the short phrase for what we're talking about here. So if someone's looking at a biochemical pathway and they say, yeah, that meets redox balance, that means you have this situation going. And so microbes will do this. And of course, this is also the process of evolution and natural selection. It depends where they're living, what kinds of ingredients, nutrition aspect they have around themselves. But very often you'll end up where they'll create a situation where hydrogen gas, H2, is one of the products that they'll then excrete. It's just, it has evolved many times in many different environments where that's just a handy way to dispose of the electrons. They'll make hydrogen gas and it works. And so very often you'll see hydrogen gas formed during fermentation processes. You'll often see acetate also formed. So that's a favorite of microbes. They'll create acetate to balance the electrons, do the redox balance. And so you'll often see that as one of the products. But having said that, remember, there's a lot of different fermentation pathways. But if you are a microbe and you're producing hydrogen gas, that is just a waste to you. It's just, you made it so that you could do redox balance, you get it out. Well, then all the microbes around you really like you because you're releasing hydrogen gas into the environment. And that is a really good electron donor for other nearby microbes to take in and use. And so the creation of H2 is actually a good thing for a lot of the neighboring microbes that love H2, because H2 loves to give off electrons. So the hydrogen gas can be very handy for other microbes. So all of this is really, if you follow the rule, do that redox balance, and then you look at what's the payoff, it's ATP production. All right, so this is also showing some fermentation processes. And obviously, we're not going to go into this in any detail, but one main thing I want to get across is, yes, we talk about glycolysis a lot, and we say it starts with glucose, but just be aware you can have a lot of other molecules that you start fermentations with. It doesn't have to be a sugar or glucose. So you can even see organic acids come in, amino acids, all kinds of other molecules can come into these pathways and create fermentation processes. And again, it just depends where the microbe's living and what is its habitat and what will be its nutrients that it'll bring in and start running these fermentation pathways. And then here's another idea. And again, this is more looking at things that we as humans use microbes for. So, of course, the brewing industry with beer and wine and so forth. But just either butter is being made, yogurt and cheeses, industrial solvents. So they're trying to hit some of the main reasons that we want microbes to ferment for us. And so industrial microbiology, we'll talk more about how microbes are being used by humans later in the semester, but fermentation is a big one, having them ferment. So here, again, just common foods that you may be familiar with, that it's fermentation. Except pickles. These days, you buy pickles off the shelf, sadly, they don't go through the wild fermentation process. Maybe the co-op has some true pickles that have been truly fermented to pickle them. But nowadays, what they do is they just put in like acetic acid and use the acid to pickle them, which isn't nearly as good. And well, I like fermented foods because not only are you getting the nutritional profile of the fermented food, but you're getting a dose of microbes too that can persist in you. So they're like a probiotic. But all of these are fermented and big industry. Yes.
When I drink kombucha, I feel like my stomach gurgles. Is that fermentation?
I think it can be. Gurgling often means gas production. And through fermentation, you do get gas production. So you'll see that, I think tomorrow and no, Thursday in lab, I think you're going to set up some fermentation experiments, and you'll actually see, we have a way to capture any of the gas production. So you'll be able to see visually that fermentation can create gas. And so that's probably part of what's going on. So yeah, kombucha is really a healthy fermented drink in this case. I've tried my hand at, do they have it here? Has anyone tried, I say kefir, but that's not really how you're supposed to pronounce it. I think it's kefir is the true pronunciation, but kefir, I've tried to make dairy kefir. I can't get it to work. It never tastes good to me, but store-bought dairy kefir tastes great. But I do water kefir. Has anyone heard of water kefir? It's some of the same microbes, but you add it to water that you add sugar to, and they ferment the sugar. And you might think, well, that's a lot of sugar. I don't want to drink something that's sugary, but because they ferment it, or most of it, what you end up with is a very low sugar drink. And so they use it, like I was trying to ferment sugary solutions, and then you can spike it with other flavors. Like then I put raspberries in for the second stage of fermentation, and it was a nice bubbly raspberry drink. I was trying to get my boys off pop, off soda pop, and so that was my way of trying to, and it worked pretty well. There were some that they like better than others, but that's what they use it for. It's a substitute for pop because pop's so full of sugar. Although I was in Sundries in the basement of the library. Have you seen their version? They call it Ollie Pop. Oh, Ollie, I don't remember how it's spelled. Ollie Pop soda cans, but the sugar content is really, really low. And then they also have prebiotics in there too. So I didn't know that they were making stuff like that. So that's another good alternative to soda pop. Okay, so what do you say, soda or pop? Soda. I say both because I know some students only hear it one way or the other when they're in their hometown. So I don't want to make sure that I cover my bases on that. I grew up saying pop. Sarsaparilla. Ooh, that's fancy. So, but anyways, okay, now let me tell you about an exception. So I just got done saying over and over and over, substrate level phosphorylation. That's what you do when you ferment. There's one interesting exception that has been studied really well. And so technically this example I'm going to show you is fermentation without substrate level phosphorylation. But I feel I do need to talk about this because this was a confusing bit of time for scientists, because when they were analyzing this pathway that they saw the microbe doing, and they ran the thermodynamics equation, figured out the thermodynamics of the pathway, they're like, this isn't enough. What's the magic number to make ATP? Negative what? Yeah, they're like, it's not negative 32. So What it was is they figured out that the microbe was doing something really wonky. It was actually tying an ion pump to this reaction, and I'll show you a picture to further explain what I mean here. But some microbes will link these pumps to this reaction pathway. And depending on the microbe, they'll either pump protons across the membrane to the outside or sodium, which is weird that you can pump sodium in a microbe like this. It's interesting. And basically, they're doing that to set up a gradient. So either a proton gradient or a sodium gradient is unusual. And then that gradient sets up, the idea of some potential energy so that when those ions diffuse back through the membrane, they generate ATP in that process. So even sodium flowing through this very special, it's called a sodium ATP synthase. And so sodium flows through it, and that's still good enough to make ATP off of that. So I'll show you a picture in a minute here, but this was the equation they were studying, and it was just so confusing. They saw the microbes could take succinate and presence of water and end up with propionate and the bicarbonate. And when they do the thermodynamics of that and look at it, it's only negative 20.5 kilojoules per mole. So that's not enough. The free energy is negative 20. You need negative 32. So it's not enough. So they're like, oh, okay, what are we missing? So, I'm sure they ran this experiment multiple times. And there's like, nope, we are not reaching negative 32. And yet we see they are making ATP. And they had this under very controlled conditions in the lab where this was the only resource the microbe had. So they knew they weren't making ATP through some other pathway. Very tightly controlled experiment. And so it took a long time. Then they're like, we got to look for some other mechanism that they're tying this to, because they are getting ATP out of this. So that's when they've hunted and found for this idea that there are protons getting pumped across the membrane. So here in this case, they're showing a sodium extruding decarboxylase, but this is your sodium pump. All right, so basically, here's your succinate mixed with water, and they're linking this that provides enough free energy. This reaction of succinate to propionate, there's enough free energy to pump a sodium. So it can do that. And if you do that enough times, then you get a sodium gradient, which then allows, when the sodium starts to diffuse back across this very special sodium ATPase, you end up making ATP. Isn't that weird? So even though that little reaction isn't enough, it's enough to pump a sodium. And if you do that enough times, you get a sodium gradient. And then you can make your ATP. So it's kind of convoluted, and it takes some extra effort on the microbes, from the microbes' point of view. So they need a fair bit of succinate to make sure this can all happen. So this is a classic example of, okay, this is not substrate level phosphorylation. You have instead, you're pumping protons to create ions, I guess I should say. You're pumping ions to create an ion gradient for your ATP. Yes.
What sort of microorganisms in their environment has this been like regularly observed in?
This one, do they say in the notes section? Usually in the figure caption, they'll say, no, it doesn't say who's doing that. So it's got to be somewhere where succinate is really heavily available. So I'm guessing it's in an anoxic environment. So I'm wondering if like sediments in a lake or river, this might be going on. But I'd have to look that up. I don't know for sure. So let's see. All right. So that pretty much is the slide set on part 1 of metabolic diversity. So this one was, kind of teasing apart what's going on with redox reactions, looking at the redox tower, reducing potential. And then we spent a little bit more time on fermentation and the whole redox balance. But now I'd like to talk a little bit more about what else microbes can do. So I'm going to shift into the next slide set, which should be, it's available, right? Okay, thank you.
So part 2 essentially. That's not what I want. All right, so not that one. Now we're going to get into a little bit more what else they can do. There's a nice quote. Life is driven by nothing else but electrons, by the energy given off by these electrons while cascading down from the high level to which they have been boosted up by photons. An electron going around is a little current. What drives life is thus a little electric current kept up by the sunshine. All the complexities of intermediary metabolism are but lacework around this basic fact. So as a plant person, I love this because they're really honing in on the idea of photosynthesis, and everything else is just lacework around that. All right, so photons of light. So we are going to talk about photosynthesis. But before we get into that one, let's think a little bit more about, they're also talking about this electron transport process. All right, so we've already covered substrate level phosphorylation, which is being shown in figure A. Figure B is more what we're gonna think about next, which is this idea, when they draw it like this, what they're trying to suggest is that you have this electron transport process going in the cell membrane of the microbe, and that allows for proton pumping. So you get a lot of protons on the outside of the cell, fewer inside, and then you get that proton gradient established, which then leads to ATP. So when they draw it like that, that's what they're trying to get at. So this is, when they draw it like this with a lot of plus, plus, plus outside, they mean, yep, nice proton gradient is getting established. And then when they show it with fewer protons like that, what they're implying is protons diffuse back in, and in that process, you get ATP, all right? So really what they're thinking about is something like this, which is a little bit more detailed, which we've gone over already. So this is the electron transport chain in the microbe cell membrane, and different microbes might have slightly different players in the game because it also depends what they're starting with, like what's feeding in electrons and what's the final or terminal electron acceptor. So there'll be different players that will be doing the passing of electrons. But really, again, the artist should have drawn that ATP synthase here, because really the goal is create a situation where you can pass electrons through this chain process. And at key steps in that process, you'll have a little bit of free energy available to pump protons. So that's what you're seeing. So here the NADH is coming in to donate off electrons and protons. The FADH2 is doing that as well. Where are those coming from? What cycle? Yep, Krebs cycle, citric acid cycle. All right, so they're donating off electrons. Protons are getting pumped across the membrane. You can see there's other areas where you get protons getting pumped. So they're trying to put it onto the scale here of the reducing potential as well. So it's just created such a nice situation where at the start of the electron transport chain, these players are really good at donating off electrons, whereas at the end, oxygen's really good at taking. And so setting it up like that allows for some free energy in this path of the chain here that can allow to, that free energy can be used to pump protons. And then you get your proton gradient, which is lovely because then you get your ATP. So if you look at, okay, microbes living in different environments, they're surrounded by different nutrients. So what are microbes doing if they don't have oxygen sitting at the end of their electron transport chain? Because there's no oxygen. They're in an anaerobic environment. Well, many microbes will choose to do a similar pathway. They still do this electron transport process. They just won't put oxygen at the end. They don't have it available, so they go to plan B. So So when you look at the phrase anaerobic metabolism, usually when they say that, they're thinking about an electron transport chain process in the membrane of the microbe. But oxygen's not at the end. So what do they put there? Well, it depends, again, on where the microbe is living and what the microbe has in terms of all the players in the donation, accepting electron game. It can be an inorganic molecule, perhaps, or it could be inorganic. No matter what they choose, if oxygen's not the terminal electron acceptor, they're not going to get as much bang for their buck. There's not as much energy released compared to aerobic respiration. So aerobic respiration means oxygen's at the very end. So no matter what they do, they're just not going to get as much ATP. However, they get more ATP doing anaerobic metabolism than fermentation. So fermentation pathways, if you're a smart microbe, that'd be plan C. That'd be your last choice. Your first choice would be aerobic respiration if you have oxygen and can use it. And then if that's not available, I'll switch to anaerobic because I'll still get a decent amount of ATP. And then if for some reason they can't run an anaerobic electron transport chain, then they might switch to fermentation. Yes. What was the chemical you were using? So oxygen. No, it has to be oxygen for the ability for it to accept electrons and get what you need to happen during the electron transport chain process. So again, like when we looked at aerobic respiration, we said oxygen takes electrons, accepts electrons and then waters the product. But again, they're not going for water. They don't really care about that per se. They just excrete out whatever the end product is. The goal they're aiming for is if I run this electron transport chain process with this starting molecule and this ending molecule and all these players, it'll set up a way for me to pump protons and get my proton gradient, which gets me my ATP. The actual final product, they don't really care about. So going back to this, if you're a microbe like E. coli, so E. coli loves oxygen. They're very happy to use it. So if oxygen is available, cool. They're going to use it as the terminal electron acceptor. Well, let's say you're running an experiment and you're mean to E. coli and you're like, I'm putting you anoxic. You're not going to have any oxygen. So if you were E. coli and you were smart, can you think of maybe something else you might want to grab and put as your terminal electron acceptor?
Iron?
Iron would be decent, and that's actually a common one depending on where you live. There's better ones out there though on this. Think about this tower. Think about what molecules really like to accept.
Like in the process of like nitrogen fixation, nitrates.
Yes, that would be an awesome one. And the reason I'm saying that, so nitrate here, nitrate's a really good option for E. coli. E. coli actually really loves to do that. If they are denied oxygen, they're like, nitrate, I can do that. Because nitrate is so close, so far down on the tower that it will get some decent ATP out of that. So the further down you are, remember, the better you are at accepting. So oxygen's the best electron acceptor, and then Fe3 plus and O3 minus. So either of those, really good. Yes. I think I know what you're thinking, and I would say generally, yes, that is a good rule of thumb, but I'm not I only minored in chemistry, so I would have to put that with, I think, not I know. But, so you see here that the Fe3 plus, the NO3 minus, they're down really close, so to speak, to oxygen on this tower.
which means they would be likely candidates if you're smart. If you're a smart E. coli, that's what you'd be hunting for. And turning on that electron transport chain process that has iron or nitrate at the end. Now, iron is very common in like a lot of natural habitats. Nitrate is something that you're going to play around with in lab. You're going to learn how this is working with E. coli and nitrate and some other microbes. So that's why I wanted to talk a little bit about nitrate here, because you're going to be running this idea in lab with a couple different microbes. And the question is, how far can they take it? So you're going to run a simple test that can give you a visual color answer. It'll change color to let you know what they did. And so let's say E. coli says, you know what, I'm all out of oxygen. Okay, I'm going to turn on the electron transport chain process that I can use nitrate at the end. And once nitrate accepts electrons, then it gets converted to nitrite. And some microbes just stop there, like that's all I can do. I don't have any other players in the game that I've evolved, so I just stop there. So you're going to test that in lab. And then some microbes say, well, I have further electron transport chain players in the game where I can just keep running and going and going. So they'll have nitrate accept, and you get nitric oxide, nitrous oxide, dinitrogen. And so these are all gases. So these will get released, and in labs, you can actually create a situation where you capture the gas. You can visually see that. But this is something that's really, really commonly done in areas high in nitrogen, like agricultural fields, for example, or forests that have a lot of decaying organic matter. And so there's nitrogen everywhere. And then you have this denitrification. So what's really going on here is you're taking nitrate, and through all those electron transport chain processes, your end waste product is dinitrogen. So what we are describing here is denitrification. Denitrification. I didn't separate those correctly. Denitrification. All right, so here, I like plants, I like growing plants. If I had this happening in my soil with my plants, I'd be pretty upset. If you've taken a plant class, you might have an inkling why plants love nitrate. That's their preferred form of nitrogen in the soil. If they can take up nitrate, ooh, they love it. They can't even touch nitrogen. Once that gets, they can't touch it. They just absolutely can't. Dinitrogen is a form of nitrogen they cannot break down. Microbes can, but plants can't. So if I'm growing a plant, especially for money, I'm trying to make a living off of this, you'd want to know if this is happening in your soil or in your hydroponic solution if you have microbes in there. Make sure that this isn't happening on a regular basis. This is not what you want in your soil. All right, here's another view of this idea. So E. coli theoretically can do this first step. Have nitrate at the end of the electron transport chain, and the waste product is nitrite. And then some other microbes just keep going. They have a longer electron transport chain process, and they'll just keep going. So Pseudomonas is famous for keeping it going. And again, it's this electron transport chain process with a different terminal electron. It's not oxygen. So here's a side-by-side comparison. So on the left, you have oxygen at the end. So this is aerobic metabolism or aerobic respiration. On the right, this is anaerobic metabolism, because you still have this electron transport chain process. You have different players here, a little bit different, but it's still this idea that you're passing electrons from donor to acceptor. And because of the energetics of that, there's key points where there's some free energy for proton pumping. And instead of oxygen, you have something else. Nitrate is very popular. Some kind of nitrogen containing compound is very popular for microbes. So how many protons are getting pumped to the outside in figure A? 4. How many in figure B? So 5 on the left, right? There's 1 here. There's 5 with aerobic and 4. There we go. 4 with anaerobic.
So they're trying to show you there's less proton pumping going on. If you don't have oxygen at the end. So that means there's gonna be less ATP. So that's how you would kind of interpret what's going on there. So Pseudomonas is amazing. So this is what Pseudomonas does. So this is the idea of E. coli can take nitrate as the terminal electron acceptor, and then the waste product is NO2. And then Pseudomonas just keeps going. So they just keep running a further electron transport chain process. And again, it's, if you are a grower of plants, you really don't want a ton of this going on. So that's why I say knowing microbiology, you can apply it to so many different fields of study. Certainly, if you want to understand plants, you have to understand microbes at some level. Otherwise, you're at a disadvantage for sure. One of those is laughing gas at the dentist. Which one is it? Is it nitric or nitrous? I always nitrous. I think it's nitrous, right? Do they even use that anymore? Do they? I haven't, and last time I had to have that, I think it was wisdom teeth pulling. Did that. That was interesting. All right, so. Did I see a hand? Yeah.
So to answer your question, would you want an organism that does nitrification instead of denitrification?
Yeah, and we'll be getting into that too. Yeah, we'll hold that thought. We're gonna get into that one. Let me just introduce this term. Chemolithotrophy. Sometimes we just shorten it down and say lithotrophy, drop the chemo off. Chemo is referring to chemicals, and it's like, well, yeah, duh, there's chemicals. And so very often we drop that off. So lithotrophy, if you look up litho, it means rock eating, to eat rocks. Trophy means to use or eat, so to speak. So this is a metabolic pathway which allows microbes to eat rocks. Sounds weird, but basically what this means is that the microbes take an inorganic source as their energy source. So what this means is they're going to pick an inorganic compound around them and they'll strip electrons off of it. And then they'll start up an electron transport chain process. So it's an electron source and an energy source for the microbe. We can't do this. We have never evolved this ability. We have to eat organic material in order to do this. Microbes, all kinds of microbes are out there that will take inorganics. So for example, hydrogen gas is an inorganic. So if microbes are like, ooh, look at all this hydrogen, I'm going to strip electrons off of that. It's easy. Hydrogen loves to give off electrons. So you grab H2, it's inorganic, and then you get it into an electron transport chain process, and you get your proton pumping and then your ATP off of that. So depending on where the microbe lives, there's plenty of microbes that will do this. They don't need organics. Compound. CPD is compound. Okay, so we'll stop there. We'll get into chemolithotrophy a little bit more next time.